First speaker today is John Irwin, who I really don't think needs much of an introduction. I'm sure most of you know John, or if you don't know him, you must certainly have heard of him. John seems to be sort of famous for me for acronyms. He works on things that have lots of acronyms. He's got DOC, ZINC, a lovely one I like called DUD, Buster, and things like that. When I asked him what he did, he said, "So I'm just a guy who does stuff." So I thought, well, stuff must be his next system he's working on. I don't know what that acronym stands for. But today he's going to be talking to us about the ZINC database. So I'd like to welcome John.
Thank you.
And hand over to him.
Thank you. Thanks to Tim for the introduction and to Alex and ChemAxon generally for the invitation to come again to this very interesting meeting. First, let me just start by thanking some people. These are some of the people that I work with on some of the products, some of the software, and some of the databases we make at UCSF. You'll recognize Erin Bolstad in the audience. She's working on ZINC. I'll talk more about what she's doing a little bit later on. Teague, in the dark picture here, is the author of the new interface of the software. And Michael and Kong have both worked on Doc Blaster, which I'll mention more than once. And Mike Kaiser did his PhD and produced the similarity ensemble approach. And the members of the lab test and help us develop the software, and the NIH supports this work. So if you're a biologist, an investigator, medicinal chemist, biologist, you're looking for small molecules, so chemistry, for your biological target or pathway or indication, so your biology. And we historically have been a docking lab. That's how we got started. And so this is a little schematic picture of docking, and this has historically been our answer to finding small molecules for biological targets. First, you start with a structure, and you start with a database of molecules, and then you use the computer to fit the molecules into the three-dimensional binding site. And the computer, of course, can do this very quickly and can sample millions and millions of conformations and poses of millions of molecules and rank them from top to bottom. And then at the end, we review the molecules that have been prioritized by the computer. We pick them out and we test them. And fortunately, we're able to routinely nowadays make predictions. And this is just an example of a paper that came out a few weeks ago from the lab against dopamine D3 receptor. And so this is a novel ligand that we predicted. So there's 40 years of dopamine pharmacology, and the structure comes out, the crystal structure comes out, and we do docking and we find things that people had never found before. So that's an example of where target-based discovery can really work, and you can find molecules that we assert, and if you read the paper, you'll see, we'll explain it in some detail. You wouldn't have found this molecule just by scaffold hopping or derivatizing previously known small molecules. And so, and this is not just a one-off. This is just a slide of some crystal structures. So we're constantly, because it's an academic lab, we're constantly testing our predictions, because docking does give you this wonderful three-dimensional prediction. I mean, we really stick our necks out and say the molecule is not only going to bind, but it's going to bind exactly like this. And we can interrogate the structure and then compare the prediction with the experiment, and we can then learn something. Ideally, even when we get it wrong, it helps us to improve our methods. But of course, I said one big caveat there at the very beginning, which was, well, you start with a structure of a protein, and whereas it's better than it used to be, there's still a lot of targets for which there's no structure. And moreover, if the question is, give me some small molecules for my target, then docking really is kind of an overkill in a way, because there's just a lot of molecules available in the literature. And recently, we've been really fortunate that the EBI in Hingston, in Europe has made this database called ChEMBL available. And this is really going towards complete coverage of the medicinal chemistry literature. And so if you cut at some level, like 10 micromolar, for instance, then you can imagine that this as being a proxy for medicinal chemistry space. And of course, that's good, except that it's not, a lot of those molecules have been made as part of a research program. You may not be able to buy them. And so we intersect the space, the medicinal chemistry space, with purchasable chemical space, which is represented by ZINC, which is the database we had to build anyway when we were going to do docking. And so this space in the middle is the interesting part. Because these molecules are biologically active, reported in the literature, and you can just go and buy them and have them shipped to you by FedEx in a week or two. And so we put together the ZINC database, and it's really just composed of catalogs from vendors from all over the world, and it's currently got about 20 million compounds, and they're not only commercially available, three-dimensional structures. We worry a lot about the representation of the molecule. So histidine is 2 molecules in zinc. Okay, it's the neutral form and it's the protonated form. You have to have both of them because the pKa is at physiological pH. And so not all databases take care of this for you, but in order to do docking, you need the biologically relevant form. So about over 100 suppliers that we use, we give it away for free, and we organize it into sets that correspond to the current opinion in the field. And it's available in popular formats that are ready to download. And there's tremendous turnover in the chemical marketplace. So we load about 10,000 molecules per day, but we reckon there's easily more than 2 million new molecules come on the market every year because the pharma industry loves new molecules because they want molecules that aren't patented yet. Okay, so there's tremendous amount of turnover. And also these companies, they make the new molecules and they make 18 months supply often, and then they disappear from the marketplace. And so ZINC is now about 8 years old. It's stable. It's widely used, widely cited, and we've recently made a new interface that's got this sort of very cool, for us, multi-level menu system. So you've got a first-level menu and then a second-level menu, so you can search by, structure, properties, vendors, all the things you're kind of used to be expecting to be able to do, and a download button and so on. And so this is the time when I'm going to pause and tell you that I wouldn't be able to show you what I'm showing you today without ChemAxon. So they're the engine behind the similarity search in ZINC and increasingly other aspects of ZINC. So this talk and ZINC itself is powered by ChemAxon. However, I'm telling you this because you're not going to be able to see it because it's at the back end, and you all know that too. And I could put Marvin up as the chemical editor, but that shouldn't impress you because most of what's going on is going on in the back end. And let me just take 2 slides to show you how we're using it and how we want to be using it. So first, we started using some other software to do this. And we've sort of retrofitted ChemAxon's software into what we already had. So we were constrained to use something that resembled what we already had. So what we did was we had an Apache web server, we had a Python and Perl backend, and then we send out the query for similarity searches to ChEMBL through Tomcat, comes back, and then we merge it into our other database, which is in MySQL. And you're saying, well, why didn't we just merge it? And it was just because we had inherited this architecture, and as a result, we have this unnecessarily complicated backend, but it works. And what we want to do is we want to greatly simplify this by using cartridge. And you all know that, and we're working on it. And what the nice thing about it is, I think with our design, you're not even going to notice from the front end anything at all. But of course, it will be much more robust and also almost for sure faster, although we don't know yet. And so Aaron, who's in the audience, is helping us do this. And it takes a while. Okay, so that's the end of the advertisement. So the one nice thing that ChEMBL with ZINC enables you to do is if you load ChEMBL annotations, so this is what? Annotations of biologically active molecules. then we loaded it into the database, and so if you go to the ZINC website, and this is online right now, you can use it, if you go to search, and then you select the targets menu, okay, well, that's not an option that's available on everyone's website. Certainly searching by properties and by structure is. And so now you can simply go to annotation and say, well, we just type in MDM, and it looks up, so within about 2 seconds, this little Ajaxy pop-up shows up and lets us select either MDM2 or MDM4. And so then when we select MDM2, we see that there are these 3 molecules that are 10 micromolar or better against Mdm2-P53, and they're also commercially available, and you can simply click on them. If you click on them, they go into the shopping cart, and you can go and buy them. So that wasn't so good, though, was it? Because there were only 3. So let's try a different target. So supposing we're interested in histone deacetylases, epigenetic targets, very, very hot right now. So we type in HDAC, and it turns out there's lots of those ones. So in this case, we can actually specify that we want not only 10 micromolar, but 1 micromolar binding, so nanomolar, really. And what that means is that they're going to be even better, and sometimes there's spurious data in the low micromolar region. And so we select HDAC3 human, and we select there, and then we get this sort of output that looks like this, where each molecule is in a tile format. We have multiple renderings, so there's multiple report formats, which I'll show in a minute. If you click on the molecule, they go blue, indicating that they've been put in the shopping cart. And so up here you can see that we've added 3 molecules to the shopping cart. You can add everything. You can just say add all, and it'll add everything on the page to the shopping cart. And if you hover the mouse over here, it brings up a nice little summary of the vendor that sells it, little buttons that quickly let you look for analogs. The physical properties, remember I said that each molecule can be represented as multiple forms, so the protonated and unprotonated forms, and links to additional information. And so this is sort of nice. And then so now under formats, this was the overview format, and then you can see all these different report formats. So if you want these structures in SDF, you just simply select SDF, and it will download an SDF file to your hard disk. Or if you want the Mol2 format or the SMILES, or if you want tables of properties and so on, you can just download them like that. Okay. There's the button. Oh, right. So now I'm going to click on Add All. And I'm going to add everything on the page. And in fact, I think there's about 30 molecules. So we're going to add them all into our shopping cart. And then when we go to the shopping cart, there's a purchasability button. I've left out one slide here. And so we've done is we've organized all the molecules in the shopping cart by the vendor that sells the most of them. So it turns out one vendor sells 16, and the next vendor sells 10 of them, and so on. This page is very long. I can't show it all. And so if you click on this link, you can either click on individual links to just take you to the vendor website to order that one compound, or if you click on this email inquiry, it just goes and loads it right into Gmail and says, "Dear customer support, would you please quote us on these compounds? We want 10 megs," and so on. And then you just press go. And so this is sort of the 0th level of integration with vendors. The next level would be to send an XML packet of all of the compounds you want to purchase right into their shopping cart. And surprisingly, some people want to do this, but a lot of them are very resistant to developing an API that would allow for direct shopping basket to shopping basket communication. And I'm not quite sure why. But sometimes, remember with Mdm2-P53, we had this problem that we only had 3 compounds. And so sometimes the medicinal chemistry space for any one target may not intersect with purchasable chemical space. And so then what do? Okay, you can't do what I've just been showing you, which is to just look up molecules from the literature. However, we've developed a method, and you probably all have your own particular favorite methods, but we have ours. It's called the similarity ensemble approach. And the next 2 slides illustrate what this is about. It's been published in Nature Biotech 2007 and Nature 2009. And really what we do is for each compound, or in the case of the paper, we did it with drugs to be more interesting for the reader. But for each compound, we compare it. We say, does it look like the target? Does it bind to the target? And the proxy we use then is that we use all the ligands that bind to a particular target as the description of the target. And so saying, does this molecule bind to this target is tantamount to saying, does it statistically resemble the ligands for that target? And so we do this we have these sets of ligands from ChEMBL, and we can have them from any place you've got associations of target ligand associations. We can compute this similarity, and then we can compute the expectation that randomly chosen molecules would also have some level of similarity. And then we normalize by random expectation, giving us a z-score. And so we're able to score that we can tell the difference between a significant and an insignificant comparison. And so we can make predictions of the biological targets of any molecule using this method. And so we've done it against all of, I think at the time I did it, there were 13 million purchasable compounds in my library, and against, at the time I did it, about 2,500 targets annotated in ChEMBL, for which there were at least 5 ligands of 10 micromolar or better. And this is just, so you might wonder whether it works, because you could imagine reasons why what I just showed you might not work. And so we were obviously very concerned about that too. And so we did some tests, retrospective tests. And what we did was we picked drugs that were already in man, and we predicted their biological targets. And sometimes we noticed that we predicted targets for drugs where we could find no reference, no information about that in the literature. And we actually managed to find for 19 drugs that were on the market, we found 32 targets that we could not find in the literature. We purchased the compound, we tested it, and so what you're seeing here in this diagram is drugs. DMT isn't really it's an endogenous metabolite. It's also a drug of abuse, but nevertheless, it's a drug for our purposes. Paxil, Prozac, and so on. And so these are the existing relationships. So this is pharmacology before we got to it. And then the lines that you're going to see up here are the new connections between drugs and targets that we predicted, and then we tested experimentally. And in some cases, the affinity for the target we predicted was higher than the patented and marketed application of the drug. Did you see the lines up here? Let's go back. Oops. There, those are the new connections that, and this information was all out there in the public, and we predicted them and tested them. So this gave us some confidence that the predictions made by the similarity ensemble approach, C, have some credibility, or at least are worth investigating. And so that's why we're able to make it available through the interface. And so C predicts over, now I've got 20 million. So I'm not sure whether it's 13 or 20. I think this represents the 2 different versions that I've done. But about one quarter of all chemistry that you can buy has at least one prediction. And you might think, well, is that high or low? And you think these compound manufacturers, they're trying to make compounds that they want you to buy. So actually, it's good that it's at least 1/4 of all those molecules have some prediction. And so now we've got, in addition to medicinal chemistry space, whether it's tangent to the curve or intersecting, we've got this newer enlarged region, which is predicted protein-ligand interactions using the C method. And so now we're interested in this little slice here. And so you can come to our interface. Now, some of my colleagues have decided that this predicting the biological targets of millions of molecules might have some value. And so you actually have to log into our website in order to see this. If you remember when we went to this page before, you only saw these pop-ups on the left-hand side. And now here under C prediction, we're going to type in MDM2, and now we say MDM2, and we select it there. And okay, and now instead of only seeing 3, which were experimentally reported ligands, 10 micromolar or better, you're now seeing dozens or hundreds of molecules where we've predicted them to be. And they might not work. In fact, quite a few of them probably won't work. But if you had to screen a library, you might want to start with some of these compounds as chemical ideas for seeing whether some of these compounds will bind. And so really that's what we're in the business of doing. If you can get it off the shelf and you need a chemical tool, go and buy it. If there are no chemical tools that you can purchase right away, at least here's a starting point. So we're narrowing it down from 20 million to a couple of hundred to get you started. Okay? And so then again, this little pop-up that shows you where you can buy it. And I just want to point out, it's a sort of a fun thing. People keep saying, oh, well, you know, your database is out of date, and people are they want very, very high purchasability rates. And so here, if from certain vendors, and one of them is VITAS-M, one of them is Molport, and when this page is built, so live, it actually contacts the server and collects via an XML RPC transaction the number of milligrams in stock and then inserts it into the page. And so if you come back half an hour later, this number may have gone down. So it's actual live information. And we're trying to do that to more and more vendors. And then we have a molecule detail page. So there's just everything is listed, including all the predictions and so on. And of course, this has all been a graphical user interface, but lots of people want to have a programmatic interface. And so if you want, for example, to look for ligands for histamine H1, for instance, that are for sale, and you want them in SMILES format, then there's just a URL you can compose, and it's very easy to see. We've got a wiki page that explains how to do this. So you just go to our website and ask for format SMILES. We want a single reference, PH7, and there's the UniProt code, and then the purchasability means, is it going to, I think purchasability 1 is 2-week supply, and purchasability 2 is make on demand, so up to 10-week supply time. And imagine you're interested in, I just want a ligand for every target. Then you can just write a little C shell script, and so you just get a list of all the SwissProt codes that there are, and then this is just a bunch of, just to make it easy so that it all fits on the page, but basically we just do a for each loop, and we for each SwissProt, we just download the SMILES and store it on our disk. So here you have commercially available ligands for every target that's available in the MedChem literature based on current purchasable information as reflected in ZINC. And so, how am I doing for time? Who's my chairman? It's okay? It's okay. Okay. Well, actually, I must have talked a little fast, so maybe there's going to be time for questions. So in conclusion, we're in the business of trying to discover small molecules for biology, whether it's for a starting point for discovering for a MedChem program, or chemical tools for biology, just to try to tease apart biological pathways. And of course we do docking. We still believe in docking. It's very much part of our DNA in the lab, and we'll continue to be interested in docking. But in many cases, many of our colleagues, and ourselves included, we just want to be able to get our hands on small molecules we can buy. And so we've developed a new interface of ZINC that makes it really, really easy just to intersect MedChem space with purchasable space. And we've implemented one system of predictions, which is our own. And you could imagine doing it with, of course, every molecule I showed you, right? So those Mdm2-P53 ligands, you could have done a similarity search using the ChemAxon backend and just gone and found, are there any analogs? So the traditional sort of SAR by catalog. And of course, ChemAxon in the form of JChemBase enables that kind of search, and it's incredibly fast. That's why we use it 3 or 4 seconds to search 20 million molecules. And so I think I'm going to stop it there and just thank you for your attention and take any questions. Hi, Wendy.
A really stupid, simple question. Why 12? Why ZINC 12?
Oh, because the previous one was called ZINC 11.
Then I have to say, why was the previous one called ZINC 11?
Because the previous one was called ZINC 10. Right. So there's a company called Enamine based out of Kiev, and they have a database of 15 million molecules that they claim they can make. They claim 70% synthesis success within 10 weeks. And so that is incorporated into ZINC. However, we have colleagues who have had bad luck against that database. And so you can choose with the purchasability flag whether to include those or not. But yeah, so we have sort of two levels of purchasability. And I think there's about 5 or 6 vendors, major vendors that sell virtual catalogs, make-on-demand catalogs. Yeah. You know, that's not my area. We're really in the business of molecules that bind to proteins. And so we have not annotated our database for that. You know, Steve Boyer might be one to talk to about that. I think he's thought of.
Yeah, because the way probably tended to move away from what you have.
So the way we think about it in the lab is what we're trying to help people with is discovering new chemical starting points. And almost for sure, you're going to have a hit to lead progression. You're going to go through ADME. You're going to end up changing that molecule so much, and then along the way, ideally, you're going to get yourself into a new chemical space. And therefore, don't worry about it too early. But maybe that's wrong-headed. Yeah.